All right, folks, we're going to start chapter 4, part 2, and this information is on exam 1. And like I said earlier in our previous lectures, it would be a great idea for you guys to make a chart in which you discuss both the structure and the function of these various cellular organelles that we will be discussing today. So all eukaryotes have a nucleus, and that's something that makes them different from prokaryotes, as we had discussed in our previous lecture. And the nucleus is the place of all genetic information, aka it's where the DNA of the cell hangs out. And most eukaryotic cells possess only one nucleus, and the nucleolus is the region where the ribosomal RNA synthesis takes place. So something you guys should know is this big thing is called the nucleus, and then within the nucleus is a region called the nucleolus. And basically, this is where DNA is made into RNA. And we'll discuss a lot more about that later on in this course. The nucleus contains the most important info in the cell, and so it needs extra gates. That's how I like to think of it. And so what it does is it contains a nuclear envelope, which is literally its own membrane. So it has a 2-phospholipid bilayer or membrane, and also has nuclear pores. And I think of the nuclear pores kind of as like doors in and out of the nucleus, and not just anything can get past these doors. It's like you need a security clearance or a special badge to get in and out of the nucleus. In eukaryotes, the DNA is divided into multiple linear chromosomes, and chromatin is just a chromosome that has protein on it. So if you see the word chromatin on your exam, we're talking about chromosomes that are wrapped around proteins. So your nucleus has a nuclear envelope, which is just a membrane around the nucleus, and it consists of nuclear pores, and these nuclear pores are to help things get in and out of the nucleus. So like I said, nuclear pores are scattered over the surface of the nuclear envelope. These pores are like gates that allow molecules into and out of the nucleus, and they are basket-like structures. So here we have a nucleus, and you see these little dots? These are your nuclear pores. And I think of them as a basket. So you can see this pore, and then things enter and get caught before entering the nucleus and get sorted. There's also something called nuclear lamina, and these are fibers that give the nucleus structure and shape. So if you wonder why the nucleus is always round, it's due to the nuclear lamina. So DNA is packaged into chromosomes, and chromosomes contain chromatin. So the DNA molecule stores genetic information. The DNA is stored in the nucleus as chromosomes, and when the DNA interacts with proteins, here you can see some of these proteins, these yellow things, it's called chromatin, and chromatin can condense down into the chromosomes, as you can see in this picture. So you have your double helix, and then you see your double helix getting wound around these proteins, and these proteins condense, called a chromatin fiber, and these chromatin fibers will then condense into a chromosome. What you need to understand is we have to pack them so carefully because we have over 3 meters of DNA in our cells. The next organelle we're going to be discussing are ribosomes, and ribosomes make proteins. Made of proteins and nucleotides called RNA is what a ribosome is, and they have 2 subunits. They have a small and a large subunit. So please know that ribosomes contain nucleotides and protein, and they consist of a large and a small ribosome. They are also the cell's protein synthesis machinery. They're found in all 3 types. They're found in all cell types and all 3 domains. So ribosomes are in prokaryotes, they're in eukaryotes, and they are in archaeas. Ribosomal RNA is what makes up a ribosome. So ribosome is ribosomal RNA and protein. And protein synthesis also requires other types of RNA. It requires something called a messenger RNA and a transfer RNA. And the way I like to think about this is your messenger RNA is the recipe for the protein, and tRNA carries the amino acids or the ingredients to the ribosome to make that protein. Ribosomes can be found free in the cytoplasm, and they can also be associated with internal membranes like the endoplasmic reticulum, which we will discuss shortly. So within your cell, you have something called an endomembrane system, and this is a series of membranes throughout the cytoplasm. And what it does is it divides the cells into compartments where different cellular functions occur. So you may have the breakdown of products in one part of the cell, and you might have another part of the cell where things are being built. And the way we do this is through that endomembrane system. Remember, prokaryotes don't have an endomembrane system. This is very specific to eukaryotes. So part of that endomembrane system is the endoplasmic reticulum, and this is the largest internal membrane in the cell. I repeat, the endoplasmic reticulum is the largest internal membrane of the cell. And it has a couple different layers. So you see these membranes and they kind of all fold on top of each other. And they have something called the cisternae, and that's the outside of the membrane. And then you have the lumen, and the lumen is the inside of the membrane. So outside is the cisternae and inside is the lumen. And the endoplasmic reticulum, or ER, is where proteins and lipids get made and processed in the cell. So you can see these ribosomes on this endoplasmic reticulum. This is where proteins would be getting made then. So like I said, there are two types of endoplasmic reticulum. So you have rough endoplasmic reticulum, and they're rough because they have these ribosomes attached to the endoplasmic reticulum, which gives them this rough appearance. And this is where the synthesis of proteins to be secreted in or out of the cells is going to be made. And then you have something called the smooth endoplasmic reticulum, which you can see here. And the smooth endoplasmic reticulum, or smooth ER, doesn't have many ribosomes bound to it, so it looks smooth on the outside. And it has a variety of functions. It's really important for making carbohydrates and lipids, but it also stores calcium and is involved in the detoxification of the cell. And the number of rough ERs to smooth ERs, that ratio can change depending on the cell type. So, like we said before, the rough ER, or rough endoplasmic reticulum, it's a flattened disc with a bumpy surface due to being covered with ribosomes. And proteins are synthesized in the rough ER are usually destined to leave the cell, so they will be secreted out of the cell. Proteins in the rough ER can be modified with carbohydrates, and when a protein is modified with a carbohydrate or covered in sugars, it's called a glycoprotein. So here you guys can see these alpha helices. Remember our primary and secondary structures of proteins. So you see your alpha helices and your beta turns. These would be the protein, and then the carbohydrate or sugar covering it is right here. So this is called a glycoprotein. The smooth ER, as I had said before, is smooth because it lacks those endoplasmic reticulum on the outside, and it's important for the synthesis of carbohydrates and lipids. Steroids and hormones are also made here, and it's also important for storage of calcium. And calcium, we'll learn later, is incredibly important for your cells to be able to talk to each other or signal. So here's just a picture showing you. So right outside the nucleus, where all that DNA is found, we see our rough endoplasmic reticulum in blue, and then the green, which is a smooth endoplasmic reticulum on the other side. So any mRNA, remember that's the recipe that gets made, could end up on the ribosomes of the rough endoplasmic reticulum where proteins will be made. And then it will go through and get processed through the rough endoplasmic reticulum and leave the cell eventually. Or if the cell wants to make carbohydrates or lipids, those will be made in the smooth endoplasmic reticulum. The next part of the endomembrane system, another organelle, is the Golgi apparatus. Now these are flattened stacks of interconnected membranes that we call Golgi bodies. And their individual stacked membranes are called cisternae. So this would be like one cisternae, but then once they combine together, they are called the Golgi apparatus. And the Golgi apparatus functions in packaging and distribution of molecules synthesized at one location and used at another within the cell or even outside of it. And I like to think of the Golgi apparatus kind of like as the UPS. So they will make sure that the protein or carbohydrate gets its finishing touches, and then it will send it to its correct destination. There is something called a cis and a trans face. So cis is going to be the side of the Golgi apparatus that is going to be receiving the proteins or the carbohydrates. And the trans face we think of as like the shipping side. So this is when these proteins are done or these carbohydrates are done, and they will then leave the Golgi apparatus and be sent to their final destination. And the way that they leave is through vesicle transport. So these little vesicles will bud off of the Golgi apparatus and send these molecules to their destination. So here's just a picture of the Golgi apparatus. You can see it here in the yellow, and you can see all of these stacked membranes. And these are called cisternae. And remember, the cis face is the receiving side. So this is the side that will receive proteins or carbohydrates. And then once that protein or carbohydrate goes through the Golgi apparatus to get its finishing touches, it will leave the trans face and a vesicle and go to its final destination. So, if we talk about protein transport in the cell, proteins are made by ribosomes. So, if a protein is made by a ribosome in the endoplasmic reticulum, it will leave the endoplasmic reticulum by a budding vesicle. So, you see here this little piece of endoplasmic reticulum buds off, and once it buds off, it's called a vesicle. And then what happens is this vesicle is going to travel to the Golgi apparatus, where it's going to enter the cis face of the Golgi apparatus. And what it will do is it will travel through the Golgi apparatus where it will get its finishing touches, and then it will leave the trans side of the Golgi apparatus via a vesicle and will finally be delivered to its destination, in this case, outside of the cell. Another part of the endomembrane system is called the lysosome. And I like to think the lysosome is kind of like the recycle center of the cell. So this is a membrane-bound digestive vesicle, and it's kind of like I said, the recycling center of the cell. It arises from a Golgi apparatus. So what happens is a bunch of vesicles from the Golgi apparatus will fuse together, and they will contain different enzymes. And these enzymes catalyze the breakdown of macromolecules. So they will break down break down proteins to amino acids. They will break down carbohydrates into individual monomers of sugar like glucose, and they will break down lipids into fatty acids. They can also destroy cells or foreign matter that the cell has engulfed by phagocytosis. So, lysosomes are the recycling centers. They break down old organelles and recycle them to monomers. And basically what they're doing is they make room for a new organelle, but they also digest other cells that were engulfed by the mother cell. And they contain digestive enzymes in the lysosomes, and they are activated by an acidic pH. So once the pH in the lysosome drops, then those enzymes will go to work breaking down whatever macromolecules are inside them. And like I said before, lysosomes are formed from vesicles budding off from the Golgi apparatus. So if I ask you where lysosomes come from, you should know that they are formed when a bunch of vesicles from the Golgi apparatus basically fuse together. Mitochondria is another cell organelle. It's found in all types of eukaryotic cells. It's sausage-shaped organelle, and it's the size of bacteria. What's interesting is that the mitochondria have an outer membrane. They also have an inner membrane, and they have an inner membrane that has a cristae and a matrix. So it's a very complex organelle. On the surface of the inner membrane and also embedded within it are proteins that carry out oxidative metabolism. And that's just a fancy term for making ATP. So if you see oxidative metabolism, I'm asking you the process in which ATP is made. And remember, ATP is what powers the cell. So those are the little molecules that hold all the power. Mitochondria do have their own DNA. If you did not know this, you get your mitochondrial DNA from your mother. So if you ever are taking a paternity test or have received one, they will look at your mitochondrial DNA to figure out who your mother is. And DNA encodes genes for specific proteins used in the mitochondria to make ATP. So here again, you see the mitochondria. They also have their own ribosomes, which is pretty neat. So they have an outer membrane and an inner membrane. They have an inner membrane space. They have this thing called the crista or cristae, which is just the space in between. And they have their own DNA and ribosomes. So the mitochondria, the cell does not produce a new mitochondria every time it divides, which is interesting because most cells will make their own organelles. Instead, the mitochondria will divide into two when the cell divides. That's what makes the mitochondria different from other organelles. And endosymbiosis is this theory that some eukaryotic organelles evolved by symbiosis arising between two cells that were each free living. So what scientists think is that one cell, probably a prokaryote, so a bacteria, was engulfed by and became part of another cell, which was the precursor for modern eukaryote cells. And that bacteria is what eventually became the mitochondria or chloroplast. So it's this idea that a precursor to the eukaryotic cell was like, ooh, hey man, I see you're making your own energy here, or ATP. Why don't you come hang out inside me, and I'll protect you from any issues like being like prey. And so what they started was a symbiotic relationship where the bacteria would make energy that this cell could use, and this bacteria could be protected from prey and environmental stresses like cold or hot. Another part of the cell you need to understand is called the cytoskeleton. This is a network of protein fibers found in all eukaryotic cells. It supports the shape of the cell and it keeps organelles in fixed locations. So it ensures that your nucleus stays in the same place in the cell or that your endoplasmic reticulum stays in the same place. It's a very dynamic system, so your cytoskeleton is constantly changing and forming and disassembling. So your cells will build up and break down parts of the cytoskeleton constantly, particularly if the cell has the ability to move. So there are three types of fibers, and we're going to go over each one in detail. The first one is microfilaments, the second one is microtubules, and the third is intermediate filaments. And what you should see is microtubules are probably the biggest, microfilaments are the smallest, and intermediate filaments are in the middle, right? So that's how I remember these. So microfilaments, also known as actin filaments, these are two protein chains loosely twinned together, and the movement is like contracting or crawling, and I'll show you a picture of that in a minute. Microtubules are your largest of the cytoskeletal elements, and they contain an alpha and a beta subunit, and they facilitate movement of the cell and materials within the cell. So if you need to move something from one end of the cell to the other, you're utilizing microtubules. And then intermediate filaments, those are between the size of actin filaments and microtubules, hence intermediate filaments, and they're incredibly stable. They're usually not broken down and they're the most durable. So I always think of those guys as like the stainless steel beams that you see in buildings because they don't really move and they don't break down. So actin is the first one, and that's a cytoskeleton element. They have long fibers made up of two protein strands, and these strands do have polarity, so their ends are different. And so you can see here these actin monomers, and what they do is they get together. And you see a positive and a negative end, and that's due to its polarity. And these monomers get together and make these two strands of actin. And remember that actin is movements like contraction, crawling, or pinching. So if you see a cell crawling along a slide, it's using its actin. Another cytoskeleton element is microtubules. These are hollow tubes made up of two subunits, an alpha tubulin and a beta tubulin subunit. And they form near the center of the cell and radiate out. So you think of it as like near the nucleus, you'd see a lot of microtubules radiating out. And they facilitate movement of cell and movement of stuff in the cell. So those vesicles you saw earlier, they actually travel along microtubules. They kind of make the highways for all these different vesicles. And then finally you have your intermediate filaments, and these are your most durable filaments. They're twinned together in an overlapping arrangement. And they're very stable. They do not break down. And a great example that I expect you to know on your exam is keratin, which makes up your hair and your fingernails, is made up of intermediate filaments. So here you can see microtubules are the biggest. And then next down is the intermediate filament. And finally, actin is the smallest. And here you can see again. So you see these microtubules going through the cell. You see these actin filaments hanging out on the cell too. And then you see these intermediate filaments. And these are all giving the cell its various shape. So what should you know from Chapter 4? You need to know who discovered the cell, who came up with the cell theory, and what is the cell theory. You need to be able to compare and contrast prokaryotes and eukaryotes, as well as telling me what are the things that all cells must have. And then you need to be able to describe the structure and function of the following parts of the eukaryotic cell. Parts of the endomembrane system, the nucleus and nuclear envelope, ribosomes, and what are the various RNA that you find in the cell. The endoplasmic reticulum, comparing smooth and rough, the Golgi apparatus, the lysosome, the mitochondria, and be able to compare and contrast the three types of fibers that make up the cytoskeleton. Good luck on your exam, and please email me any questions you might have about this material. Thanks.